We are now moving to the last, but not least, presentation on this track. Please join me in welcoming Dr. Erin Piazza, lead scientist for gene expression panel development at NanoString. The title of her talk is Introducing the Encounter Tumor Signaling 360 panel. Dr. Piazza, I'm now turning the presentation over to you.
Hello, everyone. It's a pleasure to be here today virtually with you. My name is Erin Piazza. I'm a senior bioinformatics scientist at NanoString. And today it's my pleasure to introduce you to our newest oncology panel, the Tumor Signaling 360 Gene Expression Panel. Over the years at NanoString, we've generated a number of different oncology panels trying to answer all of the various questions we can imagine people asking in the field. When we started this, we started with some simple targeted questions around pan-cancer pathways, signaling from the most canonical cancer signaling pathways. And then we looked at progression, those pathways that relate to metastasis and the tumor microenvironment. And we also created pan-cancer immune profiling to take a deep dive into the immune response to cancer. More recently, we've created some more targeted panels like our CAR-T characterization panel, looking at T-cell health, both pre and post-infusion for CAR-T products, and the metabolic pathways panel that looks deeply at the metabolism behind cancer and the cancer microenvironment. However, we've also created some more general and broad tools looking at a broader array of biology to answer questions in oncology. And those panels we call our 360 gene expression panels. The definition of a 360 panel is the 360 or comprehensive coverage of the tumor, the immune response to the tumor, and the tumor microenvironment. We've created 3 such panels over time, the first of which we called our Pan-Cancer IO 360 panel. This panel looks again at these 3 areas of biology, but focuses most prominently on the immune response to cancer. After that, we also created our Breast Cancer 360 panel, owing to our deep relationships in the breast cancer space through development of Prosigna. The Breast Cancer 360 panel again focuses on all of the biology, but primarily on the tumor and specifically breast tumors. And then most recently, we've created the Encounter Tumor Signaling 360 panel to focus on the biology of the tumor, but do so in a way that's more generic for all solid tumor studies. And this is the panel that I'll be talking about for the remainder of our talk here. So the reason why we believe this tool to be worthwhile to develop is that targeted therapies continue to be the focus for drug development. You can think about PARP inhibitors, BRAF or MEK inhibitors, IDH1 therapies, and so on. And we believe that it was time to create a tool specifically to be a companion for these sorts of studies. And as an example of that, back in 2015, when we created our Pan-Cancer Pathways panel, we started with some of the most canonical pathways in cancer biology. This list should come as no surprise to many of you and includes MAP kinase, PI3 kinase, the cell cycle, and so on. Things that are important and have been known to be important in cancer for a great number of years. However, in 2018, as many of you may remember, the Cancer Genome Atlas Consortium came out with a series of papers that highlighted some of the best results from their studies over the years and from the Cancer Genome Atlas data set. One such paper focused on oncogenic signaling pathways in cancer. And as you can see, they published a number of these pathways, and many of them were not even included in that Pan-Cancer Pathways panel, owing to the fact that oncology as a field continues to move, and the number of pathways and diversity of pathways we consider for targeted therapies continues to develop and evolve. And so for developing this panel, we started with our best-in-class panel development framework process. This process starts with foundational scientific frameworks, at least where they exist. Fortunately, in oncology, we can rely on the Hanahan and Weinberg hallmarks of cancer, which, as you know, is kind of a foundational and complete framework for thinking about questions in oncology. We can also then integrate state-of-the-art knowledge, up-to-the-date publications and other resources in the scientific literature that help us make sure that all of the content is up-to-date and informative. And then we engage our research community resources, things like public databases, high throughput data, key opinion leaders, our customers, working groups, and so on. And then finally, when we're done putting together our panels, we always make sure that they integrate well with our nCounter advanced analysis software. This software uses the pathways that we embed into the product to give you an understanding of pathway regulation and give you scores that help you understand how the biology is moving in your data set. And then we also have a feature called our cell type profiling. Our cell type profiling is embedded in the Tumor Signaling 360 panel and many of our other panels, and gives you a relative abundance of various immune cell types in your sample. And this is particularly powerful, especially in oncology, where such a large fraction of samples are in FFPE and thus cannot be subjected to cell type sorting or other methods of cell type abundance scoring. So for this particular panel, we started again with the literature search, making sure we covered all hallmarks of cancer and cancer-relevant pathways. That gave us 80 different unique sources for genes that included databases, literature, and published signatures. Overall, we considered over 5,000 genes, and these were annotated with respect to pathways. And then we used our internal development software to assess the most informative genes in each of those pathways within the context of TCGA, with consideration in some cases for tumor type specific pathways in a fashion I'll explain a bit in a few slides. And then finally, we always take feedback from our key opinion leaders to make sure that we've included all of the content and genes that would be relevant for general studies. So in the end, we have a comprehensive 780 gene list covering tumor signaling, the immune response, and the tumor microenvironment. And so in owing to our goal to incorporate biology relevant to targeted therapies, we used this particular resource to try to cover a number of the target genes from FDA approved and developing targeted therapies. These include targets like STAT3, HER3, and so on, just to try to make sure, again, that this tool is the most up-to-date and relevant tool for developing novel targeted therapeutics. Some of the unique aspects of this panel include pathways relevant only in specific cancer types. That includes pathways like estrogen signaling relevant in breast, ovarian, and uterine cancers, androgen signaling in prostate cancer, and then the NRF2 pathway, which has become of key importance in lung cancers. We also covered a number of different tumor antigens that are themselves the focus of some targeted therapies. Things like prostate-specific antigen, CCAM, GM2, which of course is not a gene, but it's the enzyme that produces it can be targeted, and then mucin 16. We also have a very comprehensive breadth of coverage for cancer-relevant pathways. This includes everything from checkpoints to metabolism to stemness and canonical cancer pathways. And then we also added some novel pathways, at least for us, in our oncology panels. Pathways like NF-kappa B, immortality and stemness, and the Hippo pathway, which is a novel pathway investigated by many people for its method of integrating both cell size, cell cycle, and the tumor microenvironment. And then finally, we included our 18-gene tumor inflammation signature. The signature measures a peripherally suppressed adaptive immune response, and I'll talk a little bit more about the development of that signature and its role in the data analysis for some of our other 360 panels in some coming slides. We also use some unique sourcing and annotation for this panel. It's always important, especially in cancer, where pathway components can be mutated or deleted or otherwise misexpressed, to measure the components of these important pathways themselves. However, it's also important to measure genes that are transcriptionally regulated by these pathways in order to measure the up or down regulation of pathway activity itself. And so to try to get at that, we included for consideration a number of signatures. These signatures are published in the literature. A few of them are listed below. And these signatures have been, again, published to measure pathway activity specifically, and thus were considered for inclusion in the panel. We also used the full TCGA solid tumor dataset for most pathways, but with the exception of a key few. These included the estrogen receptor signaling panel, estrogen receptor signaling, of course, which is only important or only has signal in breast and uterine cancers. And then further, the androgen receptor, which we focused on prostate cancer, as most of the signal for that pathway as well is only going to be seen in prostate cancer. And we wanted to make sure that we selected the genes that were going to be most informative in those specific cancers where they have relevance. And then finally, we spent quite a bit of effort to produce slimmer annotations for maximally informative pathway analysis. As many of you know, the nature of these pathways is such that many pathways traffic in and through each other during the course of signaling, especially pathways like MAP kinase and PI3 kinase that act as sort of generic signaling components in a number of different pathways. The problem with that is that if any of those genes are up or down regulated and they touch a number of different pathways, you may get spurious or less specific results for certain pathways of interest. And so we took efforts to, again, slimline these pathways to ensure that the genes in each were the most central and the most relevant to make sure that your pathway analysis results in the end are going to be extremely informative. And then, as always, we included our cell type profiling feature. As I mentioned before, this feature quantifies the relative abundance of each of these 14 immune cell types. This is a method we developed a number of years ago. A colleague of mine, still at the company, named Patrick Danaher, published on this, and it's been used successfully for years, again, for quantifying these immune cell types in the context, especially of FFPE tissues, where you don't have many other options for cell type quantification. So in summary, our tumor signaling 360 panel is a 780 gene expression panel covering the tumor, the microenvironment, and the immune response. As always with our panels, you have the option of customizing with up to 30 additional genes of interest for you. The panel itself has 40+ cancer-relevant pathways, including targets of FDA-approved and developing therapies. It also includes our tumor inflammation signature and is present in both human and mouse versions, just in case you have either human samples or you're working on some translational mouse studies. And so with that panel said, we're going to talk next about some case studies. Now, unfortunately, this panel has only been on the market for a few months, and as such, there's no data as of yet that we're at liberty to discuss from this panel. But owing to the similar biology and nature of our 360 panels, the next two stories I'll tell you are from our pan-cancer IO 360 and breast cancer 360 panels that will give you an idea of the kind of biology and results that you should expect to get from the tumor signaling 360 panel. So first, I'll tell you the story about how the tumor inflammation signature was developed. It's not just a great story to demonstrate the power of this signature, but also to demonstrate the possibilities of what kinds of signatures and information you can get out of nCounter profiling. This work was done by Merck in combination with their efforts around their anti-PD-1 drug pembrolizumab. They started with FFPE tumor tissue taken from patients prior to receiving the drug. RNA was extracted and analyzed on the nCounter platform. Depending on the custom product they were working with, they got either anywhere from 400 to 600 some genes of information out of their, and they used that data to try to discover genes and signatures associated with anti-PD-1 response. So in developing their signature, they started with the initial data set and defined and validated an initial signature in melanoma. They continued to validate and refine that signature in head and neck, gastric cancer. And then when they had an excellent candidate for their gene expression profile, or GEP, they generated the final score based on a penalized regression model in 9 different solid tumors. And then finally, that gene expression profile validated versus PD-L1 IC and PD-L1 unselected population. The final 18 gene tumor inflammation signature was published in a paper shown here. It's an excellent read if any of you are interested. The tumor signature, as I mentioned previously, quantifies a peripherally suppressed adaptive immune response. One of the things about this signature that's unique and helpful is that it actually contains very much target genes. As many of you know, in developing signatures, you don't always end up measuring genes that look related to the biology you're trying to measure. But in this case, and owing to the fact that the tumor inflammation signature was developed from targeted panels that were already focused on the biology of interest, Merck was very quickly able to get to an informative and biologically meaningful signature for response to pembrolizumab. The signature is a weighted linear sum of genes and that cutoff is able to determine whether a tumor is immune hot or cold and predicts likelihood of response to pembrolizumab and many other checkpoint therapies. In addition to the tumor inflammation signature, pan-cancer IO360 was designed with a number of other signatures in mind. Many of these are related to the immune response, as that's the primary focus of the panel, but a few of those are also related to the tumor itself and to the microenvironment. The tumor signaling 360 panel has that tumor inflammation signature and all of the immune cell type signatures that are shown on the right. But overall, none of these other signatures have been covered, albeit the biology is very much still covered for many of these biological areas. So in our first case study, this was an IO360 profiling experiment on a multi-tumor cohort from the Certum Institute in Paris. There was a heavily pretreated population of patients from a variety of different tumor types. The initial results are shown here. All of the items on the right-hand side of the graph are various cell types and signatures from the IO360 panel. And you can see that these signatures and cell types end up clustering the samples into 3 primary groups: those that are immune cold, immune intermediate, or immune hot. This is sort of a common feature of many studies where we can very easily segregate the tumors using this data into at least 2 or 3 categories based on immune status. When we did a gene level analysis of the data, we found that a number of genes included in the tumor inflammation signature were associated with objective response. Here you can see STAT1, IDO1, and CXCL9, again, all components of the tumor inflammation signature are associated with objective response. Also, in a signature level analysis, a number of different signatures, including TIS, were associated with objective response. First, you can see tumor inflammation signature itself, highlighted in the bold box, but then there were also a number of other predictors, including NK cells and Th1 cells, both of those signatures included in tumor signaling 360, and then a few key immune relevant genes like IDO1, PD-L1, and PD-1, all of which, again, are included in that tumor signaling 360 panel as well. You can also see here that overall survival and overall response rate is significantly associated with the tumor inflammation signature score. Again, this is something very commonly seen in our studies with tumor inflammation signature and multiple checkpoint blockade studies. And then one of the other helpful features of the IO360 panel is a single sample analysis that's possible through a specific fee-for-service data analysis that we do with that panel. In this case, you can see the tumor inflammation signature score for this particular single sample is high, and thus we would have predicted response to checkpoint blockade, but unfortunately this patient did not respond. However, what you can see on the right is a number of different signatures for this particular sample plotted against the population of samples in this study. And what we can see is that even though PD-1 expression is relatively intermediate in this particular sample, B7H3 expression is high, and maybe the cause or the explanation for why this particular patient, albeit having a immune hot tumor, failed to respond to checkpoint blockade. And so now we'll switch gears and talk next about our breast cancer 360 panel. It's important first to understand that quite a bit of the biology included in the breast cancer 360 panel is offered by this PAM50 signature. The PAM50 signature has as its goal to break these samples into 4 main subtypes of breast cancer. These are luminal A, luminal B, HER2 enriched, and basal-like. Each of these various subtypes has associated biology with it, both pathway biology and then biology that's predictive of the kind of treatment that should be employed for each subtype. And so in this study, the study was called the FIN-X TNBC study. It is published, and we were fortunate enough to get access to the samples from this study. It was a multi-center randomized phase 3 trial in triple-negative breast cancer, which, as many of you know, is one of the most aggressive forms of breast cancer. And patients were treated with a novel chemotherapeutic agent called capecitabine. There were 111 samples from this cohort, some of which in the TX plus CEX cohort, these patients were treated with standard of care chemotherapy plus capecitabine. And then the T plus CEF cohort, these patients were treated just with standard of care chemotherapeutic cocktail. All of these samples were run with the BC360 panel and an additional panel plus option that included biology related to capecitabine specifically. And since capecitabine is related to fluorouracil, they included quite a bit of that biology as well. So in the initial results, we saw, just like we saw in the previous study, that BC360 successfully clustered the samples into an immune hot and immune cold cohorts. And these cohorts, as you can see in the bottom, were clustered based on various signatures, or otherwise known as metagenes. And these signatures were supplemented by some of our cell type profiling. Again, the same cell type profiling and the tumor signaling 360 panel. Again, we see these 2 key biological groups, both the immune hot and immune cold, or depleted and enriched. And the anti-tumor immune activity is what clusters those various samples. We also saw that the tumors in the immune depleted subset were distributed across the various TNBC subgroups. So in addition to PAM50 that does the initial subtyping, the panel also includes additional triple negative breast cancer subtypes. And we see that these subtypes do not necessarily predict immune hot or cold tumors, which is definitely of interest because we know that subtypes in breast cancer predict quite a bit of the biology of those tumors. However, in our results, we saw that there were a number of key immune markers associated with regression-free survival in these patients. First, IDO1, PDL1, and PDL2, all genes in the tumor signaling 360 panel, were significantly associated with regression-free survival in the patients treated with capecitabine. And we also saw some additional markers associated with improved regression-free survival in these patients, and that included the endothelial cell signature and the mast cell signature. Again, the mast cell signature is present in the tumor signaling 360 panel as well. And so in summary, the tumor signaling 360 panel was designed to look at the comprehensive biology behind the tumor microenvironment, and the immune response, focusing on tumor biology and primed for studies investigating targeted therapeutics. Our other 360 panels have already, and as you have seen, shown great promise in uncovering mechanisms of resistance to various targeted therapies. And we look forward to the kinds of studies and data that will be produced on our new tumor signaling 360 panel. With that, I'll thank you for your attention and take any questions.
Thank you very much, Dr. Piazza, for a very informative presentation. Few questions for you also. Which of the 360 series panel that NanoString has would be best suited for the study of hematologic malignancies?
Great question. Our 360 panels generally think about those three areas of biology: tumor, microenvironment, and immune response, primarily in the context of solid tumors where there is a solid tumor microenvironment. However, in some hematological malignancies like lymphomas, there may actually be a solid tumor microenvironment. And so in that case, we think that the Pan-Cancer IO360 panel is the best panel, especially because it focuses primarily on the immune response, and the immune biology and tumor biology in those tumors is intricately linked. However, in some hematological malignancies, particularly in leukemia, it would be worth taking a look at our Pan-Cancer immune profiling panel that focuses pretty much solely on the immune response and would be an excellent choice for looking at the comprehensive biology of hematological malignancies, especially in cases without a solid tumor microenvironment per se.
Thank you. Can one receive a 360 data analysis report with the tumor signaling 360 panel, like for other of your panels?
Great question as well. So for the 360 panels, both Pan-Cancer IO360 and Breast Cancer 360 have a specific data analysis report. We did not produce one of these data analysis reports specifically for tumor signaling 360, and that's because we believed that the biology of that panel was well served by our existing pathway analysis. However, in the case of people who want to assess the tumor inflammation signature, there is a specific analysis report that can be generated just for that signature, and that is something that is compatible with our tumor signaling 360 panel.
Thank you. How does the content from the tumor signaling 360 panel align with the content in the Cancer Transcriptome Atlas that is available through the Genomics?
The Cancer Transcriptome Atlas is truly an exciting new product, one that I'm very much looking forward to seeing increasing amounts of data on. It definitely covers very similar biology to the tumor signaling 360 panel, and as such, there is quite a bit of overlap in the content of both of those products. So certainly if you're interested in tumor signaling 360 panel, please take a look at Cancer Transcriptome Atlas for DSP Genomics. We very much look forward to studies potentially on paired samples from both of those soon.
Thank you. Does this panel need a standard for the calculation of the tumor inflammation score?
Yes, it does. The tumor inflammation signature does require a panel standard. That panel standard can be used also for batch calibration and longitudinal studies, but certainly is required for accurate TIS calculation.
Thank you. How does the NanoString data analysis software work?
Our NanoString data analysis software starts in NSolver. NSolver is a relatively simple tool that reads in the proprietary data analysis formats that come off of our instruments and puts genes into a format that then can be transferred to our more advanced analysis software. That software is written in R and has a unique wizard tool that you can use to select some various options for your analysis. It imports the data from NSolver and then with the parameters given, generates pathway scoring, cell type profiling, gene set analysis, differential expression analysis, and many other helpful figures.
Thank you again, Dr. Erin, for the talk and the discussion. This concludes the program for the biomarker discovery track. Thank you again to all the speakers for their informative presentation and discussion. As a reminder, the speaker will follow up with any question you have submitted via email. And I want to thank you all for your participation.